Hello, my name is Amanda Smythers, and I'm a third-year PhD candidate in the lab of Dr. Leslie Hicks at the University of North Carolina at Chapel Hill. And my North Carolina space grant funding has helped me with my ongoing dissertation research, which is unraveling the mysteries of tardigrade stress response. Tardigrades are a really unique model system. You've probably heard of them because they're also called water bears, but also because they've been in the news almost constantly the last few years. Tardigrades have been shot out of a bullet and found that they could survive this type of impact. We have found that tardigrades might survive a planet-wide apocalypse. They have accidentally crash landed on the moon, and people are using tardigrades to try and save global vaccine supply through their inherent properties of certain proteins. Tardigrades are currently on the International Space Station, where NASA astronauts are conducting genetic research to see how they can survive long-term space stress. Outside of the news and the scientific worlds, tardigrades are also very popular on TV. You may have seen a tardigrade in the movie Ant-Man, as he is traveling through the quantum realm, or if you watch the new Star Trek series, you probably saw a tardigrade there. So we are really excited by tardigrades, not so much because of Ant-Man, but because of what all of these survival abilities might tell us about their inherent protein properties. So tardigrades are microscopic invertebrates, so they're usually about 200 micrometers long. They have a full nervous and digestive system, but they lack respiratory or vascular systems, and instead have these storage cells, which look like these little gray globules. And these actually can travel throughout the cavity of the water bear in order to provide nutrients where needed. They have a brain that has two hemispheres, much like ours, and eye spots on either side, so they're able to see and find their food as needed. Now here you see this is a tardigrade moving, so it has these 8 legs. Here in the center is its stomach. It just had a nice algae snack, which is why it's green. And so these are just really, really cool organisms to study. And they're easy to study, they're transparent, so we can do a lot of interesting microscopy. And in my lab, we've been working on trying to understand how they work on a proteomic level and trying to characterize that response. So the secrets, or maybe not so secrets, of tardigrade survival is cryptobiosis. So this is an ametabolic state that can last from a couple of hours, even up to years. So the longest a tardigrade has survived, and that's been empirically reported, has been 40 years frozen in Antarctica. So when tardigrades go into this cryptobiotic state, they go from this active hydrated state. You see it's about 200 micrometers. This guy is, all sprawled out. You can see all of its limbs, to what we call a ton. So during tons, tardigrades retract their limbs inward and shrink down to about a quarter of their actual size. Most tons are accompanied by a total water loss or desiccation. And yet, once again, they can survive this state for years even, with minimal metabolism taking place. So throughout the duration of my NASA space grants, I've been working to understand the different types of cryptobiosis that occurs and being able to track these on both a physiological level as well as a proteomic level. And you can see that data here of just looking at it through microscopy. So we have been able to induce anoxybiosis, so survival of oxygen deprivation, cryobiosis, this is survival via after freezing, chemobiosis, so exposing tardigrades to chemical stressors, osmobiosis, so really high osmolarity, such as 600 millimolar of sucrose, and hydrobiosis, which is the traditional method of cryptobiosis via complete desiccation. So now that we have mapped these overall cryptobioses, we can move on to answer some really large outstanding questions about tardigrade survival. So first, we want to understand, how do tardigrades actually sense and respond to distinct stimuli? How do they survive really high levels of reactive oxygen species? And to what extent does tardigrades survival rely on redox signaling? And how do these phenotypes, other than those images that I show you, differ across different cryptobioses? So we'll be doing this moving forward through biochemical characterization, reactive oxygen species quantification localization, measurements of cysteine oxidation, as well as differential global proteomics. So with that, I would like to thank the North Carolina Space Grants for their generous funding of this ongoing work. I'd also like to thank the Hicks Lab at UNC Chapel Hill and our collaborators in the Derek Collings Lab at Marshall University in Huntington, West Virginia. Thank you.